Every great scientific breakthrough begins with seeing the world differently. At SiriusXT, we've reimagined how scientists see inside living systems, right down to the architecture of individual cells. We built the world's first laboratory-based soft X-ray microscope, a tool that brings the clarity and power of synchrotron imaging into the hands of everyday researchers. This is the story of how we're changing the way science looks at life. In biology, structure determines function. The way molecules, organelles, and cells are organized defines how life works and how it goes wrong in disease. But the tools we've traditionally used to study structure have limits. Light microscopes can't see deep enough. Electron microscopes require slicing and staining that change what we're trying to observe. At SiriusXT, our mission is to empower scientists with technology that shows biological structures exactly as they exist, fully intact, fully hydrated, and in 3D. Because when we can truly see the structure of life, we can understand its function and start to change its outcomes. Soft X-ray microscopy has existed for over 30 years, but only at huge synchrotron facilities, where a beam of low-energy X-rays produced by an electron accelerator can illuminate biological samples. These facilities are rare, expensive, and heavily booked, meaning only a handful of scientists could ever use this powerful technique. SiriusXT has transformed that reality. By miniaturizing the synchrotron light source, we've created a compact tabletop microscope that delivers the same imaging power right inside a standard research lab. It's a leap that puts world-class imaging capability within reach of any institute, anywhere in the world. So why soft X-ray tomography? What makes it so unique? First, it provides natural contrast. Biological materials, lipids, proteins, and nucleic acids each absorb soft X-rays differently, creating detailed images without any dyes or labels. Second, it captures whole cells and tissues in 3D. There's no slicing or sectioning, so you see the complete structure exactly as it exists in life. Third, it delivers high resolution, down to tens of nanometers, and does this non-destructively, preserving the sample for further analysis. Together, these features make SXT the only imaging modality that combines scale, resolution, and native state integrity, a true window into life as nature intended. This image shows how the SXT-100 works as a standalone microscope, revealing every organelle inside a cell, from the nucleus to the mitochondria, in stunning 3D detail. Because the system includes integrated fluorescence microscopy, researchers can easily identify specific molecules or labeled structures and then explore those exact regions using soft X-rays. It's fast, intuitive, and perfectly suited for cell biologists who want to see both what's happening in the cell and how the cell is built, all in one instrument. Soft X-ray microscopy is also proving invaluable in the field of nanomedicine. It can visualize how nanoparticles, drug carriers, or viral vectors enter cells, move within them, and interact with key organelles. Because SXT preserves the cell in a near-natural state, it shows exactly where these particles end up, confirming whether a therapy has reached its intended target. This makes it an ideal tool for researchers developing new drug delivery systems, helping them validate and optimize treatments faster and more precisely. Soft X-ray tomography also plays a vital role in correlative imaging workflows, bridging light and electron microscopy to create a complete picture from whole organism or tissue biopsy to molecular scale. A typical workflow might start with fluorescence microscopy to locate an approximate region of interest that can be extracted from the sample, then use soft X-ray tomography to non-destructively image these sample slabs in order to precisely identify regions of interest. Finally, a thinned-down sample can then be moved to a cryo-electron microscope for atomic-level analysis. SiriusXT's microscope makes this integration seamless, allowing scientists to navigate from large-scale cellular context right down to nanoscale structure, all while preserving the same sample. It's imaging across scales, brought together in one coherent story of biology. SiriusXT is redefining what's possible in cellular imaging, and transforming how we study disease. It's a world-first technology that brings synchrotron-quality imaging into the lab. It enables whole cell and tissue imaging, capturing life in full context. It provides native state imaging, revealing cells as they truly exist, fully hydrated, unstained, and undisturbed. It's correlative by design, connecting structure with molecular activity in a single workflow. And it's transforming disease research, giving scientists faster, clearer insight into how diseases develop and how treatments work. By opening this window into the cell, SiriusXT is helping accelerate the discovery of new therapies and advancing the global effort to understand and improve human health.